One critical question after we've looked at the structure of DNA is, what does it do? It's a giant set of codes. It carries information. Those codes are for proteins that carry out multiple functions required by living organisms. But encoding proteins is not all DNA does, especially in higher organisms, where coding sequences can make up only a small percentage of the DNA. There are also regulatory sites that control which part of the code is active. DNA encodes RNA of many different types. Some of those are involved in making proteins, others are not. The DNA in bacteria, unlike eukaryotes, is almost completely devoted to the informational role, with a much smaller amount involved in regulatory processes, perhaps because these organisms are not as complex. So let's look at these roles of DNA. Only 1.5% of human DNA codes for proteins, or transfer RNA and ribosomal RNA that are involved in making protein. Lots of DNA sequence does not encode genes. There are introns interspersed with coding sequences that are taken out when a protein is produced. There are regulatory sequences that determine what parts of the DNA are available for being copied or used, and how DNA is packaged. There's unique non-coding DNA, a significant percent, and we don't know necessarily what it does. There is repetitive DNA, about 15%, but almost half of our DNA is repetitive DNA that includes transposable elements and related sequences. These transposable elements are DNA segments that can move, and we don't know a whole lot about this DNA and what function it performs. But what we are learning is that probably much of this is important in some way. Lots of different things that are part of our DNA. There's a tremendous amount of variation in the size of DNA for organisms. Humans actually are not the largest genome, though they're the largest one shown on this slide. Our genome size is about 3.2 billion base pairs, and the number of genes is around 25,000, although that number continues to be revised. Mice follow with 2.6 billion base pairs. And as we go down the list, bacteria at the bottom, they have a much smaller genome. 1.8 billion base pairs is one of the smallest genomes in bacteria, and E. coli, which is prominent in our guts, has an estimated DNA size of 4.6 million base pairs. And you'll see that there are fewer genes, although not as few as you would expect, just based on the proportion to the DNA size. And that's because the gene density is much higher in prokaryotes than in eukaryotes. In this middle range, there's tremendous variety in the number of chromosomes and in DNA size. Again, humans don't have the largest DNA, and we also don't have the most number of chromosomes. But our DNA sequence is constantly changing. We'll see when we talk about DNA repair that the sequence must constantly be maintained. Alterations in the base sequence can impact the protein that it produces. On the left of the image is a normal fruit fly. Its antennae are normal. But if you change just one base pair in a gene that's involved in developmental processes of flies, you end up with a leg in the place of antenna, which is shown on the right. So just a single base pair change can have an impact. It's very important to pay attention to what the sequence is and what it means. Also, selection of specific properties can drive the survival of distinct species. It can also have an impact within a species. An example of this is in antibiotic resistance. If you have an original population with mixed degrees of antibiotic resistance, when you grow it in a condition where antibiotics are present, you immediately select for those that have higher levels of resistance, because you're killing off those that have lower levels. So selection can drive which organisms or species survive under certain conditions. A common question is, where does our DNA come from? We've got sequences in our DNA that can be traced to Neanderthals. Recent technology has advanced so that we can get DNA from anthropological specimens. In this example, we examined Denisovan genome that was recovered from a tooth in a cave in Siberia. They were able to recover DNA, and then you can find those genes in populations across the world, with the highest densities in Southeast Asia, Australia, and some in South America. But they are also found in Europe and in North America and across the Asian continent as well. Interestingly, they're not frequently found in Africa, which may say something about when these sequences developed. So we have Neanderthal, we have Denisovan DNA, and there are probably other ancestral DNAs as well. There's a crucial role for regulating DNA expression, and we can get a sense of this by looking at the DNA sequence of human and chimpanzees, which are about 98.7% identical. If you look at which genes are expressed in the liver and the blood, the genes expressed are similar for humans and chimpanzees. But if you look at what is expressed in the brain, at least a subset of the genes that are expressed have a 5-fold difference in brain cells when comparing human and chimpanzee expression, suggesting that there are, in fact, essential roles that regulation can play to get higher or lower amounts of specific proteins, and that those differences can account for differences in organisms or behavior. If you look at the length of DNA in base pairs from the slide earlier, can you calculate the approximate length of DNA in bacteria and in humans? How do they compare? Remember that the rise per base pair is about 0.34 nanometers. Now think about what this suggests, keeping in mind the dimensions, the size of the cell. Think about what you know regarding the structure of DNA that might be used in controlling gene expression. It has to be specific. You have to be able to recognize specific sequences in DNA.